Yeah. Okay, everyone, so we have about 20 people in the room and about 30 people online. So it's really lovely to see you all for the start of semester. Room people, can you hear me okay? Great. Zoomies, can you hear me okay? Maybe let chat, fantastic. And before I start the talk, I want to open with a land acknowledgement. Well, I should introduce myself. So I'm Georgia Mason. I'm Director of the Campbell Centre for the Study of Animal Welfare. And for those of us in Guelph, as I'm sure you know, we live and work on the traditional territory of the Mississaugas of the Credit, one of the Anishi-Nabek peoples. And so we live in the purple area up here, round about here, which basically means we live on land that was purchased by First Nations led by Mohawk Chief Joseph Brant in 1792. And even like me, you feel like you're really still very much learning about Indigenous history and rights and the sort of darker sides of Canadian past, then I'd really recommend the website of the Mississaugas of the Credit because it's really good. I would also recommend going along to the University of Guelph's events on Friday to mark Orange Shirt Day or Truth and Reconciliation Day, in which flags will be lowered to half-mast and we'll be remembering many thousands of parents and children who were split up from each other, and that was, and worse, that had lasting legacies that still are in effect today. And then the other thing I'd recommend to really put a local perspective on this is that the Woodland Cultural Centre is a local museum established in what was the oldest residential school about 30 miles away in Brantford, and they do virtual tours of the school and have some first-person accounts of the experiences there. Tonight, they have a tour tomorrow night. It costs $10. It's really awful, but it's also really well done, and I'd really recommend doing it. They sometimes do in-person tours as well, but they froze that for the pandemic, and they haven't reinstated them, I think because the area is basically a crime scene at this point, because they've got a 600-acre property, and they're now, searching it for bodies. So I'd really recommend, especially given the timing of this week. All right, so switching to to introduce Jessica Cait, who was supposed to be giving this talk. Jess, if you're watching this recording, it is wonderful working with you. For those of you who don't know her, Jess did an immunology MSc in UBC. I then pretty much had a bidding war with UBC trying to poach her to our lab. We won, woohoo! She won an NSERC PhD scholarship, and initially she planned to use her lab skills to look at markers of neuroinflammation in our differentially housed mice. But then the pandemic hit. She was in lockdown in Vancouver for months and months. And during them, instead, she audited a new course run by Population Medicine. Olaf Burke and Jan Sargent run it on meta-analysis. And she gradually went through that. She went through a journey that many of you go through, initially being terrified by statistics and gradually discovering that it's nerdily deeply enjoyable. And that's the situation she's in now. So it's her meta and analytic skills that result in the product that I'm going to present today. So I'm sad she can't be here, but she was brilliant helping me prep for the talk to deal with all questions, but she says she's happy to deal with questions afterwards if I'm lost. So what we're going to talk about today is how rodent housing affects the health of animals and then potentially even the research results that they generate. And this is really an animal welfare perspective on preclinical replicability and also translatability. What do those terms mean? I will come onto that in a little while. So what this talk has two halves. The first half presents some published work that came out in BMC Biology, and some of you will have heard this before, and it basically looks at how housing conditions affects disease vulnerability and mortality. And then we're going to present some new work, some new systematic review analyses, which look to investigate the effects of housing on research results and see if these can help explain the current replicability and translatability crisis. And this work's extremely new. Like some of the results came out last week, we're still processing them, but this seemed like an ideal time to get feedback. And then we'll wrap up with some implications for rodents and for other species as well. Excuse me. Okay, so most rodents around the world live in small, rather empty cages like the one shown here. How empty this is really depends on where these animals live on the planet. So hundreds of thousands live in research labs. The vast majority live in the US and China, where there are no regulations to give them anything really. So most will typically live just on bedding. They won't have nesting, they won't have shelter. This will be it. Although individual institutions may vary. Those living in Canada or the European Union are a bit luckier, and they are required to have nesting, shelter, or both, depending on the species. But it's still pretty small and barren, and it's very different from their natural habitats. So naturally, rats and mice are commensal with humans. They typically live in our sheds, our garages, our greenhouses, our warehouses, and they would then use a nest that they build themselves as a base, and they will forage for nesting material and food for tens of meters in variety in different directions. So they'll typically have a sort of cubic home range that could be hundreds of cubic meters. And this is obviously very different from the laboratory setup. And we know these differences matter because we know that many of these natural motivated. So if you ask rodents, do you want to sleep in a shelter rather than out in the open? They say yes. If you ask them, do they want to build a nest? Yes. They want to run, they want more space, they want to explore novelty, they want to dig their own burrows, they want to be able to urinate and defecate far away from where they sleep, which seems very civilized. And so what they want is often very different from what they're given. And this in turn matters, and we've known this really since the 1980s. So we've known that rats and mice will typically work very hard to reach what is often known as enriched environments where they have more resources to interact with them. So mice, for example, can push weights heavier than themselves to live in a setup like this rather than the traditional empty shoebox. And if we compare animals living like this, and I should say we've put enriched in scare quotes, I'm going to say why at the very end of the talk, but if we compare animals living in enriched conditions with animals living in small shoeboxes, we find many signs of relatively poor welfare in the conventionally housed animals. So just to give a few of many, we find that the shoebox housed animals have reduced stress resilience, so that if something nasty happens to them, like an injection or rough handling, they show a greater physiological response. Their heart rate might increase more, and they also take longer to recover, whereas the enriched housed animals just seem to shrug off nasty experiences more readily. And we also see lots of abnormal behaviors in the shoebox cages. So particularly stereotypic behaviors like bar biting, running around in circles, and barbering, where animals pluck out their own or their cage mate's fur. But despite all this, there hasn't been a kind of radical change in how laboratory rodents are kept. And so Jess and I thought, well, if all this great ethological data really isn't causing much of a change, maybe data on health could be a bit more impactful. So for a start, it might reach veterinary experts who might have some more legislative powers than the average ethologist. And so it's very well known that chronic stress arising from being in a situation that you'd rather not be in is bad for your health. We really know this very well in people. So many studies of humans show that chronic stress can precipitate a bunch of diseases and can also end your life prematurely. And it does this through a bunch of mechanisms, but including prolonged sustained activation of the HPA and SAM axes, which then leads to various costs or harms called allostatic load, which in turn then make you more vulnerable to disease. And this recognition that chronic stress can be physically harmful and promote disease was recognized by the Institute for Laboratory Animal Research in the US, which is a regulatory body that has laboratory animal oversight. And they said, when stress is this bad, we should call it distress. And so they defined distress as a situation so aversive that you see changes in physiology that then predispose animals to greater morbidity and greater mortality. Now, that's a bit vague, disease and death. Can we be more specific, Jess and I looked at the literature on human stress to identify 7 particular conditions that time and time again research reveals are stress susceptible. And these are cancer, or rather the cancers, because there's many of them, cardiovascular disease, stroke, anxiety, depression, viral infections, and asthma. And for all of these conditions, being in a state of chronic stress increases the chance that you'll get these diseases. But importantly, once you get them, chronic stress also means they'll progress faster, they'll become more severe, they'll be longer lasting, and they'll be harder to treat. And these elements in red are important because when we see these diseases in lab rodents, they're not appearing spontaneously. No one's waiting for their lab mice to get cancer. They're induced deliberately. So the induction, the onset of these diseases in lab rodents is really artificial. But the rest, how ill they get, how easy they are to treat, and how long they take to recover is entirely down to their own biology. And so these, if we're right, should be susceptible to stress. And because stress has such a wide range of just bad knock-on effects throughout the body, it also increases what's known as all-cause mortality, which is a fancy way of saying dying from just everything. Dying from stuff even we don't know what killed you. Hey, deaths are mysteriously higher in highly stressed groups. So these insights from humans meant that our broad prediction, which is that conventional cages cause sufficient stress that they compromised health, can actually be turned into some very specific testable predictions. And these are that compared to enriched housing, they should be more severely affected by the 7 stress-sensitive diseases that we've identified, and they should show increased all-cause mortality. So these 7 stress-sensitive diseases are pretty well studied in laboratory rodents. As I mentioned, they're always induced artificially by human researchers. So, excuse me. So the bottom left shows how stroke is induced artificially by messing with cerebral arteries. The top right shows how anxiety and depression can be induced by repeatedly doing things like restraining rodents in a very tight tube. The bottom right shows tumors that have been induced. I don't know how, but it could be that they were implanted, that tumor cells were injected into the animal. So to test the hypothesis, Jess conducted a systematic review and meta-analysis, and she followed some really lovely techniques aimed to maximize transparency and replicability as laid out in the Cochrane Handbook, which is like the medical Bible for meta-analysis. And so, for example, all the methods were pre-registered. We said what we were going to do before we did it, and we lodged that in the atrium. She also did a massive screen to really try and capture absolutely everything that was out there. So initially she had over 10,000 hits that she then narrowed down according to studies that met particular eligibility criteria. And I'm not going to give you all the details, but the crucial things are that we needed experiments in which both conventionally housed and enriched animals were studied in one of these diseases. And so within each of our studies, animals would be kept in a condition that the researchers called standard or conventional or control, relatively barren, and then something that they called enriched. This could just involve the addition of a shelter, as shown here. It could involve the addition of a whole bunch of things. But basically what we then have from the literature is almost a series of matched pairs, where in each study, the animals are identical in sex, species, strain, and disease, but the housing is different. And then across the studies, you've got a lot of variability because we're looking at rats and mice, different strains, and a range of different diseases, a whole range of different standard conditions, and a whole range of different enriched conditions. We just used whatever was out there. So this gives 2 examples, and I want you to imagine another 212 added to this, because Jess managed to find 214 studies buried in 186 articles. So each of these, as I said, contained a conventionally housed group and an enriched comparator group, and they spanned 5 of our 7 diseases. We simply couldn't find work on asthma nor on viral infections, and they were roughly evenly split between our 2 species, mice and rats. So just to give you a couple of sort of incidental attributes of these studies that emerged before we tested hypotheses, one notable thing was that every single study treated the bad barren cage as though it was a neutral control environment, and all of them treated the better cages as though they were some treatment group, even though the neutral controls are very, very barren. Only a third of the studies actually described their conventional cages, which means we just had to kind of guesstimate what they were from local minimum regulations. And we may have been right, we may have been wrong. 60% of the studies also group housed their animals, which is great, but nearly a third of them housed animals individually, which was a bit disturbing because that's not great for rodent welfare. Only 6% of studies used both males and females, and in the single-sex studies, nearly 70% used males only. And this represents a common skew in biomedical research to basically ignore females in favor of males, which is basically just sexism, and has led to a lot of moves to recognize that sex is a biological variable. If you don't collect data on both sexes, you won't be able to extrapolate to different sexes and genders of humans. Okay, so to test the actual hypotheses, Jess had to extract comparable metrics from all of these diverse studies. And to do this, she extracted figures from which she could calculate something called effect sizes. So effect sizes are standardized metrics of impact that allow you to compare the results of very, very different studies. Because some of these studies are measuring anxiety from behavior measured in seconds. Some of them are studying cancer by looking at the size of a tumor measured in millimeters, all different units, all different scales. And we need to make these chalk and cheese measures into comparable things. So what you do here is you calculate something called the standardized mean difference, which basically means you take the mean of one group away from the mean of another, and then you standardize it by dividing by the pooled standard deviation. And this gives you a unit-free number that's typically between 0 and 1 or 2. It can be higher, it can be positive or negative. And the numbers are typically interpreted like I've put in the little box here. Sorry, like Jess put in the little box here. So 0.2 is deemed a small effect, not much going on. 0.5 is deemed a medium effect, and 0.8 or larger is deemed a large effect. So that was for the stress-sensitive diseases. For death, Jess had to do something a little bit different. So every time a study reported mortality, and this could be any study, not just working on our diseases, she reconstructed Kaplan-Meier curves from survival curves presented in papers and used these to calculate hazard ratios, which are the relative risks of living in the two housing conditions. So a hazard ratio of 1 means enriched and conventional housing conditions are equally risky to live in terms of dying. A hazard ratio of 2, if it's conventional to enriched, would mean that conventional housing is twice as dangerous as enriched to live in. The chance of dying at any one moment is doubled. Okay, so I'm going to hop to the results, skipping over a bunch of details, and I'm going to walk you through how meta-analysis results are often presented. And they're often presented in something like this, which is called a forest plot. And I'm going to start with the simplest disease, which is cardiovascular disease, only generated 10 studies and was only quantified using one metric, which was the size of atherosclerotic plaques that developed in the aorta of affected animals. So we've got one measure here, plaque size, reported in 10 publications, and the black rectangles show the effect size and the confidence intervals generated by each study. Anything to the right of that dotted line means we've got a positive effect size, which means that conventional housing is causing more severe plaques than enriched housing. Anything to the left would mean the enriched animals are protected. This experiment found no housing difference. All the others found that enriched housed animals had smaller plaques, developed and maintained smaller plaques than the conventionally housed animals. The average effect size is shown here at 0.72, which means it's medium to large. And this housing effect was highly statistically significant. Okay, so now I'm going to walk you through the other 4 diseases, starting with one that generated similar effect size and going up to diseases that were more strongly affected still by housing. And these other diseases were a bit more complicated in that they were more widely studied and more different measures were taken. So instead of a detailed forest plot, we're going to show you just some summaries. So for cancer, there were 72 studies, and these studies measured 4 different things, and we've just coalesced summaries of the studies for each of these 4 measures. So some looked at the effect of housing on tumor weight, some looked at tumor volume, some looked at the rate of metastasis from a primary tumor, and some looked at the number of tumors that were just spontaneously developed even when a primary tumor was not implanted. So here, the middle of the blue diamond indicates the effect size for each of these measures. Once again, the red diamond indicates the overall mean, and the overall mean effect size is again small to medium, with again conventionally housed rodents developing more severe cancer. And again, this effect was highly statistically significant. Okay, so the next disease looked at was stroke. Once again, stroke, the severity of stroke is measured in a variety of different ways. We've got 6 measures here. One is a composite score of how impaired an animal is behaviorally by a stroke. Another is a more precise measure of how precisely the animal can move its limbs in particular ways. Morris-Watermays measures of spatial ability are often looked at because the lesions often compromise hippocampal function. And then there are 2 measures of just overall coordination looked at. How good are rodents at balancing on a beam, and how good they are balancing on a rod that's rotated and then gets faster and faster. So at what point do they fall off? And then some studies also look at the physical side of the size of the lesion itself, a fixed period after it was induced, which kind of gives you a measure of how destructive it was or how well the animal healed. So there were 56 studies that looked at these 6 different measures, and across these, the average effect size is 0.87. Once again, the conventionally housed animals are more severely impacted by the strokes that they're given in the research. And once again, this is highly statistically significant. And 0.87 means now we've got a large effect size. Housing is really having quite a massive effect. But the most affected diseases at all were in studies modeling anxiety and depression. So it's psychiatric illnesses that seem most affected by chronic stress. So if we look at rodent models of anxiety, here animals are genetically manipulated or treated in even more stressful ways. And then their fearfulness is measured in a variety of different tests. And the 4 key ones we had measures for were a light dark box test, which is how long an animal takes to emerge from a safe seeming shelter into an open environment. The elevated plus maze, which again looks at cleaving to shelter versus going out into the open. Social interaction test, because social interactions can provoke anxiety. And open fields, how bold are animals roaming around an open area. 28 studies looked at housing effects here. And across these 4 measures, the overall effect size was 0.91. So again, really large. And again, conventionally housed animals are being rendered much more anxious by the same treatments than our enriched housed animals. And finally, for depression, so depression, we had 28 studies looking at depression. 3 depression measures of depression-like states are recorded. One is learned helplessness, so how animals give up struggling in a particular challenging task. One is hippocampal volume, because the hippocampus tends to shrink in depression. And the other is anhedonia, so the loss of pleasure, typically looked at by rodents' motivations for tasty treats. And across these 26 studies and boiling down these 3 measures, we can see that there's an absolutely enormous effect of housing treatment here. The effect size is 1.24, which is kind of off the conventional scale. So surprise, surprise, of course, it too is highly statistically significant. So what we can see here is for the 5 stress-sensitive diseases for which we can get data, all our predictions were met in that conventionally housed animals are much more vulnerable. They develop more severe diseases despite the induction being identical. All right, but what about death? I hear you cry. So death, we found a similar result. So here, Jess and the team looked at the hazard ratio comparing enriched to conventional housing. Anything to the right of the dotted line means that conventionally housed animals are in trouble. And what Jess and her team found was that both in studies looking at disease, not just R5 disease, but any diseases, and studies just looking at spontaneous deaths that just happened, and there was about 16 of one, 20 of the other, the average hazard ratio was 1.48, which pretty much means that at any moment, the risk of death is 50% higher for an animal in conventional housing than in enriched. And this causes and mysterious causes we know, causes we don't know. Risks are just higher for animals in conventional housing. So again, this supports our final prediction that in conventional housing, we see increased all-cause mortality. And so this supports the hypothesis that conventional cages do cause sufficient stress to compromise rodent health. And in turn, this means that conventional cages then are causing distress as defined by the Institute for Laboratory Research. So we got a fair amount of publicity for this research, but not that much, because people honestly don't seem to care about rats and mice, even though rats and mice are lovely and smart and cute and delightful and sentient. The few journalists who talked to us went, yeah, but what does this mean for humans? And I'm like, but the mice, no, what does this mean for humans? And so that topic is then the next section of this talk. What could these housing effects mean for the validity that come from these rodents, and how might that matter for humans? Because if we can't get people to care about rodent health, we must be able to get people to care about human health. And we were particularly interested in this because biomedical research has, in the last decade or two, realized that it's in the midst of 2 crises, a crisis of replicability and a crisis of translatability. So replicability basically means if a scientist runs a study in one lab, and another team try and run the same study in another lab, do they get the same results? You would really hope so, because we all hope that when we do research, it's relevant just beyond the tiny confines of our own world. And yet that hope doesn't seem to be being met. So a Nature survey found that 70% of researchers surveyed reported they couldn't replicate other researchers' findings. And several meta-analyses have shown that in the biomedical research world, between 51 and 89% of studies cannot be replicated. So that's pretty amazing. So at least half of all preclinical biomedical work using animals yields results that no one can ever find again. So why does this matter? It matters because, well, I mean, all you scientists in the room and online, imagine you're reading away, trying to understand your subject matter, you're writing your lit reviews, you're designing experiments, you're writing grants. Half of what you're reading might just be mirages, ephemera that don't really mean anything. They're just type 1 errors. So that means science risks building of cards, it wastes our time and effort. But beyond that is potentially a waste of money and animals when they're used in the research. So in Canada alone, roughly $1 billion a year is spent on animal research that then no one can replicate. And this involves 2.5 million animals. And globally, hundreds of millions of animals are used in biomedical research, and half of them, at least, are generating these weirdly ephemeral, non-replicable results. What are the causes of this problem? Because no one wants this. Clearly multifactorial and really difficult. If it was easy, it would have been fixed already. It's been 3 causes have been suggested in the literature. One is that there are methodological problems, that when people run their experiments, they cut corners, they don't randomize their animals across different parts of a room, they're not blind when they collect data. And these problems mean that conscious and unconscious biases can creep in to create bogus results that don't really mean that much. There are also statistical issues. One is that sometimes analyze their data 15 million different ways in the hope of finding something. This is known as p-hacking. Students in the room, if you do this, you will find something. Just as if you read enough tea leaves, you will see a handsome man on a horse, but it doesn't really mean anything. So that's one problem. Studies are also often underpowered, which means that they often fail to find stuff that is there. And when they do find effects, they can only find giant effects, which no one can ever find again because they were flukes. There are some sociological issues as well, that we can't publish null results. It's really difficult. So we're all under pressure to find if P is less than 0.05. When we write papers, we're often encouraged to be succinct. So there's poor reporting in papers. It's hard to replicate a study if you can't really see how it was done. And underlying all of this are the perverse incentives of academia, where, as you either know or you're going to find out pretty soon, we're on a publication treadmill where we're rewarded for producing papers and not necessarily for producing. Our hypothesis is that rodent housing is another contributory factor to the replicability crisis, because if rodent housing can change behavior and health, then logically it might alter research results. At least in our data set, only a third of the studies actually even described how the animals are held. And we know conventional housing differs in what the bedding is, whether nesting's there, whether shelter's there, how big the cage is, what the cage is made of. So that means all sorts of unreported variability in the literature, which no one can see because no one writes it down, but is important for rodents potentially. And so we think replicability will be reduced between laboratories that unwittingly use different types of housing that influences their rodents in different ways. All right, but replicability is not the only problem. The other crisis is the translatability crisis, and this pertains to the problem that even when we look at research that's actually replicable, so that's under half of biomedical research, only the tiniest fraction actually proves relevant and useful to human patients. So estimates suggest that only between 9 and 14% of really promising looking animal research actually effectively translates to humans when it's run through clinical trials. So this suggests that animal research, even when it's replicable, is producing a hell of a lot of false leads that people run after. So why does this matter? Oh, sorry. And here are some just statistics. Is it getting colder and colder in here, by the way? It is, isn't it? Okay, so for Alzheimer's disease, there have been over 40,000 studies using rodents allegedly to study Alzheimer's, and not a single one has yielded an effective therapy. In stroke, over 1,000 treatments have looked like they worked in animals, woohoo, and then we run them through human trials and they don't work at all. So it's spectacular attrition. Why? Well, kind of seems bloody obvious, but it matters partly because it goes against public trust. When the public are asked, how do you feel about using animals in research that's sometimes a bit horrible for the animals, the public go, oh, well, as long as their welfare's looked after and as long as it's useful. And the Canadian public, bless them, think that about half of animal-based research generates something useful for humans. The true value is probably under 1%. 90% of animals used in disease research, which is not that nice, by drug companies are not yielding usable research. So that's and it's cost a fortune. Running human clinical trials is really expensive. And so drug companies are spending nearly 90 million a year on clinical trials that don't go anywhere. I didn't feel sorry for drug companies till I read this stuff. And now you kind of see the bind they're in. There's a grimace in the room, but seriously, it's a big problem. And this is just the problem caused by false leads. Another problem, which we can't really quantify, is that there are probably drugs that didn't work in animals, but would work in us. It's hard to quantify them because if they from nowhere, and yet we know that acetaminophen, aspirin, penicillin, also tamoxifen, the breast cancer drug, all would have failed today's animal tests had they not already got through the pipeline and been used in people. So what these missing, this suggests animal research generates false negatives as well. And so this obviously means, well, we don't even know what we can't find. So what are the possible causes? Again, in the literature, people have come up with different ideas. Some different animal species are too different from each other and from us. The whole exercise is futile. So that is one possibility. Other people have said, no, it's just these poor research practices again. Again, there's lack of blinding, lack of randomization. That just means some of our results are just caused by people's unconscious desperation to find cool results. And then several people have suggested it is something to do with animal welfare and animal housing. And applied ethologists have been suggesting this for 2 or 3 decades. So Trevor Poole, Hannah suggested that stressed rodents don't model most humans. And in recent years, some biomedical researchers have been joining in. So some have argued, yes, you know, you're right, conditions that stress animals or that compromise brain development just can't yield valid data. And others have pointed at the metabolic effect of cages as well, that animals that can't move, especially if they're rats, often get really fat. So, you know, do we want all our data to come from obese rats? And then mice without nesting materials are as cold as we are in the room. Do we want all our data to come from animals, if they're mice, that are cold? And the issue here is not that the data are just wrong, but just they have very limited external validity. So human psychologists have recently recognized that psychology data, which seeks to say something about humankind, actually only really comes from test subjects who are Western, educated from industrial rich countries that are also democratic. Those data might apply to all the humans on the planet, but they might not. The external validity might be limited. So the weird acronym was so cool. Jess and I came up with our own. Cramped, it hasn't taken off, but if you can, if you could help, that would be awesome. So we would argue that if rodents are cold, rotund, a bit forced, I know, but hey, abnormal, male biased, poorly surviving, enclosed and distressed, maybe their data then only extrapolate to some people, to lonely, stressed, overweight men who need research data, but they don't necessarily extrapolate to a happy, physically fit, you know, fulfilled women. Then how could cramped animals change research results? There's 4 different ways, and one of them people sometimes make a fuss about, but doesn't actually matter. So I want to walk you through this because then it explains the predictions that Jess tested in her most recent work. So there's 4 potential ways in which housing conditions can affect results. And I've shown one of them here in graph A. So here we have conventionally housed animals and enriched housed animals. There's a measure of disease severity on the y-axis. And then in both of these laboratories, they're being given a drug, and that's the gray groups. And you can see that the drug is successfully tackling the disease. So the animals in the enriched conditions have less severe disease because they're less stressed. So their values are lower in all the cases, but that doesn't matter because both the labs would draw identical conclusions that the drug works. Hey, we've got a drug that works. So here we've got a main effect of housing, technically in kind of stat speak. So the housing matters for the absolute values, it matters for animal welfare, but it doesn't matter for research conclusions. And both these labs are able to replicate each other's results. But we can get patterns potentially where housing does make qualitative difference, changing what you conclude. So here I've got the same setup, a lab with conventional housed animals, a lab with enriched. The gray group is the drug treated group. So in which housing condition here does the drug work? Works in one and not in another.
What's in the conventional?
Yeah, so in this scenario, the drug is working if you house animals in conventional cages. Doesn't really work at all in enriched housing. So here, the nice lab is unable to replicate the results of the nasty lab. The nasty lab is running to Pfizer going, fund our work, and the enriched housing lab is just going, oh, we found nothing. And so what this means technically is that housing conditions are interacting with drug effectiveness. We've got a statistical interaction between housing type and drug effect. All right, we could also find things flip. Oh, and here's a real life example. So those of you who think that red wine is good for you, it looks like the antioxidant effects of resveratrol in grape skins are much reduced or absent if you look at enriched housed animals. So in this study, this is a measure of oxidant effectiveness in the brain. I don't really understand it. The control and resveratrol groups differ, shoebox housed animals, but if they're enriched, the enriched animals benefit so much from the housing that resveratrol doesn't have any additional benefits. So that is like scenario B, a real life example. All right, scenario C, again, a drug only works in one housing condition. Now, where is it only working? Enriched. So here, if you're working with shoebox cages, you're like, oh, damn it, we found nothing. If you're working with enriched cages, you're like, woohoo, we're onto something. And the nasty lab is then unable to replicate the cool results of the nice lab. So here's a real life example of this type of housing drug example. So here, blocking a serotonin receptor is an effective stroke treatment in enriched animals, but not in non-enriched. So here is the delay it takes for an animal to fall off a rotating rod. The longer the delay, the better their balance. Here is the drug group in enriched animals, and the drug is having a brilliant effect. The drug is doing absolutely nothing in the conventional animals. Which best model human stroke patients? I am not sure. And then the fourth possibility, though it may be a bit unlikely, is that the direction of a drug effect just completely flips. So it doesn't vanish in one condition, it totally changes in direction according to housing. All right, gosh, am I going to get through this? That is the challenge. So Jess's question was then, does using different cage conditions, conventional or enriched, generate different conclusions about disease risk factors like sex and age and potential treatments like therapeutic drugs? And she called all of these disease modifiers, and she realized she could explore these effects using her giant trove of gleaned papers that compare conventional and enriched animals. So the predictions are, if this is true, she should find interactions between housing and the impact of these disease modifiers. So she should find patterns like my graph B, C, and D. In other words, these are the patterns that would support the hypothesis. Modifiers only acting in conventional conditions but not enriched, modifiers only conditions but not conventional, or modifiers having completely flipped results in the two housing conditions. So what did she do and what did she find? So she updated her search and added another 2,000 new finds that she trawled through, screened everything with new inclusion criteria, looking for experiments that used a 2x2 factorial design, housing cross-factored with some kind of disease modifier. And again, she extracted means and standard deviations to calculate an effect size now reflects the size of an interaction. So how this works is it's a standardized mean difference again, but Jess calculated the effect of a modifier within each housing group and then looked at the difference between them and standardized that by the pooled standard deviation. And how you interpret this is just the same as before. 0 means nothing's going on, 0.2 is small, 0.5 is medium, and 0.8 is large. Okay. So she found 70 articles containing 97 studies that used a 2x2 factorial design. Some of these were very similar. They looked at the same modifier and the same disease outcome. So those she pooled to increase their power, leading to 64 calculated effect sizes. These spanned all 5 of our diseases. And there was a range of disease modifiers. So 60% used therapies that it was hoped would tackle the disease. 35% used modifiers trying to probe a mechanism of action, and sometimes they were even detrimental. And then around 5% looked at other modifiers like age or sex. So this is what she found. So in her 64 cases, in 17 of them, the modifier effect was bigger in conventional than enriched cages, like our scenario B. And in just one of these, so the one shown in black, but not the ones shown a bit more kind of ghostly, in one of these, that was significant. In 33 of the 64 cases, the modifier effect was bigger now in enriched housing than conventional. And here, in 16 of these cases, the interaction between the modifier and the housing was statistically significant. So all these black lines are showing a significant interaction. The ghostly ones are not significant. So that's like scenario C. And then in 14 of her 64 cases, the modifier effect flipped direction between conventional and enriched houses. And in 6 of these cases, the interaction was significant. So we called this a negative effect because it's so kind of weird. And again, the black ones are significant, the ghostly ones are not significant. So there is definitely something going on, at least in some cases. So to summarize these results, in 24 of her 64 cases, so that's about 36%, the effects of a disease modifier were significantly altered by housing, such that there's a significant interaction between housing type and a disease modifier. So this means people working with different housing wouldn't replicate each other's findings. My laptop, I think my laptop is cold. So 1 out of 64 was like scenario B, 16 out of 64 was like scenario C, and 6 out of 64 was like weirdo scenario D. So does housing quality alter research conclusions? Most of the time, no, at least in her data set. In around 2/3 of the time, it didn't make a difference. But around 1/3 of the time, modifier effects did significantly interact with housing. I'm just going to skip this for time, actually. So what are our conclusions? So one set of conclusions is to do with animal welfare. Current rodent housing conditions do clearly empirically induce distress. And so we urge that rodent housing should be improved. This clearly isn't good enough. Being cramped and frustrated of natural behaviors matters for rodent health. And I think that's ethically troubling. We also came to passionately hate the term enrichment, because enrichment sounds like chocolate and Manolo Blahnik shoes and lovely luxurious extras. But these are basic, arguably basic needs that help keep animals healthy and alive. So we'd like to say a better term is resources. Pat Turner's already argued for this. We totally think she and her collaborators are right. We should talk about poorly resourced and well-resourced environments, not conventional and enriched. We suspect these findings apply to other species as well in the lab beyond rats and mice. So Michelle Lavery in my group has recently shown that if you house zebrafish with resources that they prefer, they live significantly longer. And maybe effects apply outside of the lab as well. Maybe when we look at farm animals or zoo animals kept in really quite impoverished conditions, maybe there too, we would see evidence of distress. Now, these animals aren't being used to study stress-sensitive diseases. They often don't live long enough to die a natural death, but still we could look for markers of inflammation, immunosuppression, and allostatic load. As for conclusions about the use of animals in research, supposing you don't care about animal welfare at all, and that is a depressing number of people, these results, I think, still are important because they show that rodent research doesn't always change, rodent housing doesn't always change research results, but it can. And we found interactive effects between housing and disease modifiers in around 1/3 of our studies. So if this figure is representative, and obviously that's a big if, then such patterns are widespread. Around 1/3 of results are non-replicable because of the housing conditions, and that's pretty worrying. So we think at very least, rodent housing should be better reported so that you can actually replicate how other animals were housed. It's not even in the ARRIVE guidelines at the moment. As for translatability, which quality of housing is most relevant to most humans? That's a difficult question, and it probably depends on who you're trying to model. So if you're trying to model well-off people with fabulous social networks, well-enriched, well-resourced rodents might be best. If you're trying to model people who've had really tough lives, who've been in and out of group homes, and then they're in prison. I mean, we know socioeconomic status and educational level predicts lifespan. If we want to model the most unlucky people in society, we should keep on going as we are. And if we want to model everyone, maybe we should deliberately have heterogeneity in our research setups. Okay, so just to wrap up, so what are we doing next? And if you have ideas, I'd love to hear them as well. So one obvious thing is to try and work out which enrichment, in quote, types best improve rodent health. Because this was often so poorly reported, Jess has been surveying the PIs of all the papers that she pulled together with the help of Ola, who's there. They've got all the data that they can, and analysing that will be Jess's next PhD chapter. We've been thinking about potential postdoc ideas. I think it'd be cool to repeat both meta-analyses, but looking beyond our 5 to 7 diseases, especially to look at the effects on research results and see if they apply beyond stress-sensitive conditions. And another question could be, how does social housing affect animals? Because if rodents are often still being kept on their own, then never mind physical resources, is it legitimate to extrapolate from rodents who are socially isolated? And then other future plans in my lab, we're quite interested in seeing whether there's cage side markers of distress that would be more useful than just waiting for animals to die. So does stereotypic behavior predict an early death? Does sitting there in a depressed like huddle predict an early death? Or do physical markers of aging, like this little black mouse who's become all gray, are our mice in conventional housing becoming grey faster? And are those the ones that have the greatest allostatic load? So that's the sort of thing we're looking at next. All right, so I have loads of people to thank. Jess's review team, Ola for helping with the survey, all the other students in the Mason lab, you lot in the Campbell Centre for just making this such a fun environment in which to work. I would also like to publicise, while I can, the next fall seminar, which will be on October 19th. And it will be Rowena Packer in from the UK to talk about pandemic puppies. You think it's going to be all about socialization problems, but a surprising amount is going to be about organized crime. And she is a fantastic speaker, so hope to see you there. And if there are any questions, I would love to hear them.